We just finished talking about how if you have a cup of liquid of a solution that has a certain amount of solute and a certain amount of solvent in it, and then you put a cell inside that particular cup, a little cell inside that cell, it's got a cell membrane around it. And there's a certain concentration of solute that's a part of that is for the solution inside the cell. And what we're talking about is that when the concentration of solute is different inside the cell versus outside the cell, versus in the water out here, basically water is either going to go into the cell or come out of the cell. If the water here has more solute in it, then it's hypertonic, hyper above. If it has less solute, it's hypotonic, below, lower activity level, hypothermia, that your temperature is getting too low, and so on. Now, if you put a cell in a hypertonic solution, then what's going to happen is that liquid is going to travel, that solvent is going to travel, water is going to travel out of the cell into the hypertonic environment, trying to equalize the concentration. So if this is 10% is 10% solute, and out here is 20% solute, the water is going to go from the inside to the outside, trying to turn this 20% down into something like 12%, while bringing this down to the 10% that you find inside the actual cell. Same with the other way, that if it's in a hypotonic solution, the water out here is got a lower amount of solute. So let's say it's 30% in here and 10% out here solute. Then the water is going, since the solute cannot move, the water is going to travel into the cell, because it's a hypotonic environment. It's going to travel into the cell, and then as it goes in, it's going to cause that cell to swell and perhaps burst. So if you put a cell in a hypertonic environment, it's going to shrink as water leaves it. If you put it in a hypotonic environment, it's going to swell and possibly even burst if there's enough of a difference. Those are the interesting ones. The third possibility is an isotonic environment. Isotonic environment just means that this has 50% solute and this has 50% solute, or this has 10% solute and this has 10% solute. The concentration is the same. Because there's no difference in concentration, then the water just moves across the membrane at exactly the same amount. The isotonic solution, isotonic meaning it has the same concentration, doesn't have any effect on the cells. They just stay the same if you put them in an isotonic solution. This is key to the concept of how plants wilt and recover. When plants don't have enough water, they wilt. When they get more water, when they get watered, they recover. So in its original state, the plants, they basically speaking, as they send water out through their leaves, they have less and less water inside of them. But if the soil around them is dry, it forms basically an isotonic environment in terms of the amount of water, same amount of water in the soil as there is in the roots. Now, if you increase the amount of water in the soil, it's going to flow into the roots, therefore increasing the amount of water in the roots. That allows the plant to pull that water up through the roots into the whole plant. So it's constantly stealing water from the roots to put it in the rest of the plant, reducing the level of water, so it'll pull in more water from the soil. This reaction, this way of doing things, is part of what causes a plant that when it's run out of water and is starting to wilt, you water the ground around it. What's actually happening is that now that the ground has become a hypotonic solution in relation to inside of the roots, the water starts flowing into the roots and the plant begins to recover.